The ground tissue of leaf, we don't have cortex, endodermis, pericycle, stele, all that. We will have a tissue of the leaf is called as mesophyll tissue. Okay. Now, what is mesophyll tissue? A tissue present inside the leaf which is not differentiated into cortex, hypodermis, epidermis, all that section. So, basically the tissue of the leaf is called as mesophyll tissue. In monocots and dicots, you will see the variation. So, what type of variation is there? For example, in dicots and monocots. If I draw a rough diagram of leaf, cuticle, for example, if we think that it is a rough diagram of TS of leaf, okay, in dicot leaf. So, what is present in dicot leaf? You have cuticle is present at the outer side, then you have epidermis. So, immediate to epidermis, you have a tissue, long elongated barrel shaped cells which are made up of chloroplast pigment in them. So, these are called as mesophyll tissue is again divided here into two types, that is palisade tissue and spongy tissue, right. So, in dicots, as we know, mesophyll tissue is consisting of two sections, palisade tissue and spongy tissue. So, if we think it is a TS of leaf, so cuticle, epidermis and the long barrel shaped elongated cells which are continuous without any intercellular spaces, which has chloroplast pigment in them, which are green in color, which are helpful in undergoing photosynthesis, these are called as palisade tissues. Okay. Now, round, spongy, ball like structures which are present remaining part of the leaf, and these are loosely arranged, there is a lot of intercellular spaces in between them, then those type of tissues are called as spongy tissue. Okay. In the center you will have the stellar region, vascular bundles are covered by bundle sheath. Okay. This is different story, we are talking about ground tissue. So, here in dicots, the ground tissue is also called as mesophyll tissue, as I said earlier. This is palisade and spongy tissue. So, the on the upper side of the epidermis or upper side of the leaf, below the upper epidermis, you have barrel shaped palisade tissue, and the remaining cells are consisting of spongy tissues, round sponge like, because they are sponge like soft and they are having a lot of intercellular spaces, they are not in a continuous ring. There is a lot of intercellular space between 2 to 3 spongy tissues. That is why, as they are very sponge like, they are called as spongy tissue. Now, in case of palisade tissue, there is a possibility of occurrence of palisade tissue only on the one side of the epidermis, or in some plants you will see the occurrence of palisade tissue on both the sides of the epidermis. Then when it is occurring on both the sides of epidermis, we call it as upper palisade tissue or lower palisade tissue. In between, the common tissue which is present is called as spongy tissue. So, altogether palisade and spongy tissue is altogether called as mesophyll tissue. Now, when we talk about monocots, this was about dicots where palisade and spongy parenchyma is present. Palisade tissue and the spongy tissue is made up of parenchyma, that is called as spongy parenchyma. Now, when we talk with reference to the monocots, we will not see the presence of differentiation of mesophyll tissue into palisade and spongy tissue. Like we will see the mesophyll tissue which is there made up of isodiametric cells, and with the whole tissue is made up of isodiametric cells, and these are without any intracellular spaces. These are isodiametric cells without any intracellular spaces, and these cells consist of secretory tissues. Monocots consist of isodiametric cells without intracellular spaces. These also consist of secretory cells in between them. Secretory cells are also present in the mesophyll tissue of the monocots, and in the monocot leaves, in dicot leaf, on both the sides of the vascular bundle you will see the presence of bands of cholenchyma, whereas in case of monocots leaves you will see the bands of sclerenchymatous tissue present on the both sides of the vascular bundles, and these are covered by a layer called as bundle sheath. Okay, this is about the ground tissue of the leaf, that is called as mesophyll tissue in dicots and monocots. Dicots, it is differentiated into palisade and spongy parenchyma, whereas in monocots the cells are isodiametric without intercellular spaces. In some cases you will see the presence of secretory cells. Okay, this is about your ground tissue in both stem, roots as well as in the leaf. Why we are in detail studying about the mesophyll tissue is, in monocots we said the vascular bundles are surrounded by bundle sheaths. So what is the function of bundle sheaths here? Bundle sheath consists of chlorophyllous parenchyma. So in monocots, the vascular bundle is surrounded by bundle sheath, and each bundle sheath consists of chlorophyllous parenchyma. This chlorophyllous parenchyma is helpful in undergoing photosynthesis. So in monocots, where does the photosynthesis occur? Because of the presence of chlorophyll parenchyma in the bundle sheath, you will see more occurrence of photosynthesis. So this is in detail the structure of mesophyll tissue in the leaf, I mean the ground tissue of the leaf. We have studied the ground tissue of root and stem.